I guess we should get started. We're in for quite a treat today. Not only is this the first time we've had an open forum with food, we're going to be treated with Neil's talk. So I think this is kind of a double special day today. So Neil has a great background. Moving from from Oklahoma, he moved to and went to school in Northwestern, and then transferred after finishing his BS. Moved out to one of my favorite schools, UCLA, where he did graduate work, both in crystallography and protein. And we were talking earlier today, he actually had some of the similar experiences that I did at UCLA, which was a wonderful place to work. And during that time, did beautiful work on crystallization of thermophiles in structure function. After that, he moved up to David Baker's lab, and his career has been stellar ever since. His main drive from his earlier work has been looking at questions using protein to look at novel templates, novel polymers, and make vaccines as well as viral systems. And so today, I think we're in for a real treat. And Neil's going to tell us a little bit about the work he's been doing on viruses and how to arrange particles that emulate viral systems. Get into questions of geometry, positioning, identity or specificity, and going beyond that a little bit. I think it's going to be a real treat. Again, try the pizza. Hopefully it's going to be stable now from now on. We'll have open seminars, food, and discussion. Hopefully.
Awesome. Thanks, Bruce. And I'm really excited to be here today and happy to be here in person with all of you. I think there's nothing more fun than scientists getting together and talking about science. So hopefully we can have some of that sort of fun today. Okay, so I'm going to talk today about computationally designing nanoparticle vaccines, and the stories that we'll talk about today are in influenza, coronaviruses, and then hopefully beyond. So just a disclosure, we spun out a biotech company on some of the technology discussed today. So, I don't think I need to tell anybody this anymore, but we're in the midst of a technology-driven revolution in vaccine design. This has been happening over the last couple of decades, and it's really just been an absolute sea change, as epitomized by, I would say, the mRNA vaccines for COVID, right? So I'm highlighting on this first slide just a few examples, but there's really just a deep bench of new technologies that are being applied in vaccine design that are changing the game. So the first is antigen engineering, and one example, kind of the poster child at this point, is prefusion RSVF. So using protein engineering techniques to stabilize the antigen in the conformation that is most relevant to the immune system, right? And RSV was considered a very, very challenging vaccine target in the pharmaceutical industry for a very long time. And then the Vaccine Research Center's stabilization of the prefusion form in 2013 really changed the game. And now several candidates based on that molecule are in phase 3 trials. And Pfizer's is looking particularly good. So a second example, of course, is novel technologies for delivering vaccines, such as mRNA. I don't need to wax poetic about this. We've all seen what this can do. And then finally, another example I wanted to highlight is antigen engineering for improved breadth to protect against not just one virus, but many strains or isolates of a virus or viral families. And one technique here is this concept of germline engineering, where you germline targeting, where you engineer your antigen to engage specific B cell precursors and activate and expand specific B cell lineages, and then guide them to the development of broadly neutralizing antibodies. And the amount of technology that goes into this approach is really just mind-boggling, and I think really encapsulates the revolution that we're living in right now. So one other technology that has really come to the fore in vaccine design is the design of particulate or self-assembling vaccines. And it's been known forever, for decades, that self-assembling or particulate immunogens lead to more robust immune responses. And one example of how this happens is through B cell receptor clustering on the B cell surface, where a monomeric or a soluble antigen will engage a single B cell receptor, whereas presenting a repetitive array of antigen clusters B cell receptors together. That leads to stronger signaling, activation, better T cell responses, and just an overall more potent immune response. It's not only BCR clustering that leads to this. Particulate antigens also have improved trafficking to lymph nodes in vivo, improved interactions with the innate immune system that leads to their better performance. So today we're going to be talking about self-assembling protein immunogens. And of course, this is a clinically validated vaccine modality. So the HPV and HBV vaccines that have been in use in the case of HBV for decades are self-assembling protein immunogens. They're virus-like particles because these proteins from these particular viruses have a natural tendency to self-assemble into these shapes. And these are some of the safest and most potent subunit vaccines that we have. So these ones form these virus-like particles naturally, but many antigens that you might be interested in using in a vaccine don't naturally self-assemble. And in those cases, you need a self-assembling scaffold if you want to make a particulate vaccine. And there was a beautiful paper in 2013 from Masaru Kanekiyo at the Vaccine Research Center showing that this non-viral structural protein, ferritin, which self-assembles to an octahedral complex with 24 subunits, can be used to scaffold what I'll call a complex antigen, in this case, influenza hemagglutinin, a trimeric viral glycoprotein, highly glycosylated, multiple disulfide bonds, right? This is something that you can't put on just any particulate system, right? Ferritin proved to be robust enough to handle this complex antigen. So one of the reasons that self-assembling proteins are such a useful platform is because it allows you to seamlessly integrate protein-based antigens through genetic fusion to the self-assembling subunit. So now you just make a protein and it naturally self-assembles to these particles. And because proteins fold and assemble to single low energy native states, every particle looks just like every other. So these tend to be very regular, very monodisperse, which from a manufacturing perspective is very advantageous. And so on the right is just 3 serological measures that show that the nanoparticle vaccine significantly outperforms a commercial, a seasonal flu vaccine at the same time. And so this work really kicked off a huge new area of research, and many people have followed up on designing these sorts of particulate vaccines. The papers I'm showing here are just an outdated and very selective subset. But all of this work in the field has really focused on a small number of naturally occurring self-assembling protein scaffolds. So in almost every case, one of 3 scaffolds is used: ferritin, encapsulin, or lumazine synthase. So although these have been very successful, there are cases where these scaffolds don't get the job done, right? And then you're out of luck, right? Also, the structural features of these scaffolds are fixed. Ferritin is ferritin. It's always going to be ferritin. You can't really change its fundamental structural properties. So if you want to try something different, if you have an antigen that requires a different solution, you're kind of stuck, right? And so that's the problem that we have been using computational protein design to solve. And the basic idea is that if we have robust methods for designing new protein-based technologies, we should be able to tailor them to specific applications. And so I use this kind of ridiculous cartoon to encapsulate this idea. If you wanted to invent a new technology, let's say an alarm clock, and you limited yourself to only using things that you had lying around the house, you would end up with this ridiculous, non-robust contraption, right? But this is not how human beings invent new technologies. We design pieces to spec and modularly, and then we put them together to make simple, robust, and controllable technologies, right? And so that's what we want to do with proteins. So during my postdoc in David's lab, we developed a general computational method for designing new self-assembling protein nanomaterials. This is a graphical depiction of that method. This is all implemented in a software suite called Rosetta that we use to design and model protein structures. And it comprises 2 fundamental steps. There's a docking step and a design step. So we're building these things the same way that Mother Nature does. And that is to say, by combining symmetry and non-covalent protein-protein interactions. So that if you think of like ATP synthase, microtubules, virus capsids, all of these wonderful molecular machines are built by combining symmetry and non-covalent protein-protein interactions. So here we're using icosahedral point group symmetry, which can be represented by a collection of 5-fold and 3-fold rotational symmetry axes. And then we go find, or increasingly these days, we just design from scratch oligomeric building blocks that match those elements of symmetry. We align them along the appropriate symmetry axis in our target architecture, and then we sample the rigid body degrees of freedom. In this case, there's only 4. You can space each building block in and out along its axis, and you can rotate it around its axis, but all other moves are disallowed by symmetry. So 4 DOFs is really easy to sample, and so we sample those computationally and identify docked configurations where these building blocks fit together nicely. So we're kind of like playing Legos with proteins, right? And then we go into the design step where we use Rosetta's sequence design capabilities to make up a new amino acid sequence at this new protein-protein interface. And if we get that amino acid sequence right, then we should, or it should, uniquely specify assembly to our target architecture. So at the end of the day, what you get out of the computer is a hypothesis in 2 parts, a pair of amino acid sequences and a 3-dimensional structure you predict those sequences will assemble to. And of course, because this is computational, we can do this over and over and over again. So you can make millions of candidate nanomaterials. We take those hypotheses into the lab and test them. And this is some work done in collaboration with Jacob Bale, a very talented graduate student in David's lab at the time, who designed a number of different 120 subunit megadalton scale nanomaterials with icosahedral symmetry. And you can see in the electron micrographs, again, every particle looks just like every other, so they're very regular and monodisperse. And when we calculate averages from the negatively stained micrographs, they very closely resemble projections calculated from the computational design models. So the proteins in the real world look just like what we made up in the computer. And we can go solve crystal structures of these things and compare them on an atom by atom basis to determine the accuracy of the method. And in these 2 examples and in many other examples, the deviations at the backbone level over the entire assembly are less than 1 angstrom. So we're putting every atom in these couple megadalton scale materials exactly where we said we would, with an accuracy of about half the width of an oxygen atom. And so you really can design these things with atomic level accuracy. And the implication, of course, is that this method now allows the design of novel nanomaterials with customized structures, right? So if you want a nanoparticle that's more porous, you might go for the architecture on the left. If you want a nanoparticle that's less porous, you might go for the architecture on the right. If you want a 16 nanometer, 17 nanometer, 18 nanometer, 19 nanometer particle, you can now design that. So my group over the last few years has been using these materials in 2 biological applications: biologics delivery and vaccine design. And so we're doing this the same way that Masaru and his team at the VRC did it back in 2013 with ferritin. We genetically fuse an antigen of interest to one, or in some cases both, of our nanoparticle subunits. And then we just make these 2 proteins as standard recombinant biologics. So there's nothing special. You just make a protein the same way you always do. And then when you mix these 2 proteins in a test tube or in a reactor, they spontaneously self-assemble to the nanoparticle immunogen. And so we've been doing this with a huge number of different antigens over the last few years with a worldwide network of collaborators. It's been super fun to work with everyone on this. So I'm showing here examples from RSV, influenza, and 3 different particles from HIV, but we've really explored a number of different indications here and convinced ourselves that this really is a robust and versatile technology. And so I want to go through 2 stories in detail today, one on influenza and then one on coronavirus, on SARS-CoV-2. And really, ultimately, one of the questions that we're asking and one of the questions that we're focused on now is, can we design vaccines that protect not only against a single virus, SARS-CoV-2, but against families of viruses, all SARS-like coronaviruses or sarbecoviruses. And both of these projects have been close collaborations with Barney Graham, who recently retired from the VRC, as well as Masaru Kanekio, who's still on over there, and with David Wiesler here in the biochemistry department at UW. So the idea or the concept that is underlying our approach here is this concept originally conceived by Dennis Burton in the early 2000s of structure-based antigen design, or some people call it reverse vaccinology 2.0. And the idea here is to characterize antibodies of particular interest in complex with an antigen, and then learn from those structures how to design your best vaccine. And the types of antibodies that we're interested in, if we want broad protection against multiple different viruses, are antibodies called BNAbs, broadly neutralizing antibodies. And the idea is that, many antibodies are going to bind to strain-specific epitopes, right? So antibodies B, C, and D in this figure bind to triangle, square, and rectangle-shaped epitopes, whereas antibody A binds to a circle epitope that is conserved among these three related viruses, right? So the identification of broadly neutralizing antibodies like these from humans is an existence proof that the human immune system can come up with solutions to this problem of virus diversity. The question is, can we provide the right information to the immune system to elicit those types of antibodies? And one approach to do that was, again, pioneered by Masaru, our collaborator, and we've been working together on this for the last few years now. And that idea is to use what he referred to as mosaic nanoparticles. So here, you make a nanoparticle immunogen that co-displays related but antigenically distinct antigens that harbor a conserved epitope. So here, the conserved epitope is shown in pink, and you can see that it's common to all of the antigens displayed on the nanoparticle, whereas strain-specific epitopes are represented in different colors. And the idea here is to skew B cell activation in favor of cross-reactive B cells. So strain-specific B cells will react. So this gray B cell is interacting with this white epitope, which exists only once on the nanoparticle. So this B cell doesn't see the nanoparticle. It only sees one copy of the antigen, so it's more like a soluble antigen that weakly activates that B cell. Whereas the hot pink B cell sees this multiple copies of this conserved epitope that's presented in this antigen array. And so that B cell gets the benefit of the nanoparticle, is strongly activated and expanded. So hopefully the idea here is that you are preferentially activating these cross-reactive B cells. So Masaru published this work originally in 2019 using monomeric influenza hemagglutinin receptor binding domains displayed on ferritin. And he did this just by making these genetic fusions. So there's the hemagglutinin RBD, there's the ferritin subunit, and then co-transfecting these into cells. And when you co-transfect them, the ferritin subunits self-assemble with multiple different monomeric antigens on the surface. The particles look beautiful, and then when you immunize mice and do some serology, you can see that the mosaic immunogens elicited broader neutralizing activity than a monomeric nanoparticle, a mixture of multiple different nanoparticles that displayed the same set of antigens, or sequential immunization, heterologous prime boost with those different nanoparticles. So this was the first evidence that the mosaic approach could actually lead to more broadly protective responses, in this case within H1N1 influenza. But Masaru and Barney could not display trimeric hemagglutinin on ferritin because it's a homomeric particle that self-assembles on its own inside the cell. You can't display multiple different trimers. You might get mismatching between the trimers that disrupts particle assembly. And so they came to us because our two-component nanoparticle approach allows you to make such immunogens quite easily. And so this is work led on our lab's end by Dan Ellis, who recently graduated from the group. And what Dan did is he took the 4 hemagglutinin strains that are in current commercial seasonal flu vaccines and made genetic fusions to the nanoparticle trimer subunit in gray. And those can be individually assembled with the pentamer protein to form different nanoparticles, each displaying multiple copies of a single hemagglutinin. Or you can mix all 4 of these trimers together first. Nothing happens until you add in the pentamer, at which point you drive co-assembly to form this mosaic nanoparticle immunogen that co-displays all 4 hemagglutinins. The particles look beautiful no matter whether they're monovalent with a single hemagglutinin or the mosaic. And David Wiesler's group got a single particle reconstruction of these, showing that both the icosahedral core as well as the displayed hemagglutinins were highly ordered and forming the native-like structure in terms of the hemagglutinin. And then Dan developed a series of methods. This is an IP-based method to show that we were in fact getting co-display. So if you pull down the mosaic particle with an anti-H1 antibody, you pull down all of the hemagglutinin-bearing subunits and nothing is left unbound. But if you do the same with the cocktail, when you pull down with the anti-H1 antibody, only the H1 particle comes down, the rest stay in solution. So this shows that in the case of the mosaic, we are in fact getting co-display on the same nanoparticle. So then Seyhan in Masaru's group at the VRC did a series of immunogenicity studies in mice, ferrets, and non-human primates to evaluate the performance of these nanoparticle vaccines. And we were comparing them to, again, commercial seasonal vaccines that had the same hemagglutinins in them. And what we saw when we looked at vaccine-matched responses, so the same viruses or very, very close to what was in the vaccine, we saw that both nanoparticle immunogens, the mosaic in pink and the cocktail in blue, were about the same as current seasonal vaccines, including in terms of hemagglutination inhibition. But where we started to see differences was when we looked at mismatched viruses, so looking at broader antibody responses. And here we used a panel of historical influenza viruses that Masaru's group had built and looked back in time, how well did this vaccine protect, or in this case, neutralize, viruses that had antigenic drift relative to what was in the vaccine. And you can see that over about a 15-year time span, both nanoparticle vaccines outperform current seasonal vaccines by about a log or log and a half. And so even though this experiment was looking back in time, we think that maybe this is a reasonable proxy for looking forward in time. And perhaps a vaccine like this could provide protection across multiple flu seasons. We don't know that for sure yet, but that would be the hope, is that this could be like a supra-seasonal influenza vaccine. We did mismatched challenge studies. So this is using an H3N2 from back in 1982. And we saw that both nanoparticle vaccines did a much better job at protecting mice from mismatched challenge than the seasonal flu vaccines, and that this seemed to correlate with neutralizing activity. And then we looked even further afield. We looked at heterosubtypic responses, so things like H5N1 bird flu, H7N9, distant cousins on the flu family tree. And here's where we saw real differences from the seasonal vaccines. So in many cases, you get very poor responses against these heterosubtypic hemagglutinins, but the nanoparticle vaccines actually provide pretty robust responses. And this was again borne out in challenge studies in mice, ferrets, and from passive transfer from immunized non-human primates, where we saw consistently that the nanoparticle vaccines provided much more robust protection than seasonal vaccines against these heterosubtypic challenges. And if you look across all of the challenge data, you might notice that the mosaic nanoparticle seems to have a subtle but consistent edge over the cocktail, suggesting that it is providing broader immunity. And in passive transfer from NHPs, so we immunized the NHPs and then transferred purified IG into mice, and then challenged the mice, and we saw the same thing, showing that this protection is antibody mediated. We wanted to understand the nature of this heterosubtypic protection better, and so Dan went back to the nanoparticles that we had made, and he reverted the computationally designed interface that drives nanoparticle assembly to the original amino acids at these positions to create a non-assembling version of the vaccine. So here, all of the same proteins are there. They have just a few changes in their amino acids. So all the T help is there, everything. It's just these proteins can't assemble to form the nanoparticle. And when we immunize mice with these, the data is in pale yellow. We saw that vaccine matched responses didn't really change much from the nanoparticles or the seasonal vaccines, but heterosubtypic responses dropped all the way back down to the level of seasonal vaccines, showing that these broad antibody responses were directly linked to formation of the nanoparticle structure. And so to get a better sense of what was going on there, we thought that these antibodies might be directed at the stem of the hemagglutinin molecule because there's a highly conserved epitope there that is targeted by many known broadly neutralizing antibodies. And so when we measured binding, serum antibody binding to antigens that only comprise the stem of H1 or H3 hemagglutinin but lack the head, we saw dramatic differences between the nanoparticle immunogens and seasonal vaccines. And David Wiesler's group performed polyclonal epitope mapping by EM. So they processed polyclonal serum antibodies to Fabs and then formed complexes with antigen and visualized these polyclonal antibody antigen complexes by EM and saw that complexes formed with a vaccine matched hemagglutinin targeted multiple epitopes on the antigen, both the head and the stem, but the responses were primarily against the head, which is expected. The head region of hemagglutinin is known to be immunodominant. But when we formed complexes of these polyclonal Fabs with a heterosubtypic hemagglutinin from H5N1, we observed only stem-directed antibodies. And so this shows that the protection, the heterosubtypic protection that we observe is mediated by stem-directed antibodies, some of which, this major class of which, closely resembles the human BNAb, MEDI8852. And so it looks like these mosaic nanoparticles are eliciting BNAb-like antibody responses. And so this vaccine is now in a phase 1 clinical trial at the NIH. So we are eagerly awaiting the data out of that trial. So the second story is on SARS-CoV-2 and coronaviruses more broadly. And this, of course, was in response to the pandemic, starting about 2 years ago. And Lexi Walls, absolute powerhouse research scientist in the Wiesler lab, and Brooke Fiala, a powerhouse at the Institute for Protein Design, took this on and led the team that rose to the challenge here. And initially, we displayed both the pre-fusion stabilized spike trimer on our nanoparticles, as well as just the receptor binding domain. The RBD nanoparticles actually perform better in our hands than the spike particles, so that's what we took forward. So again, just a genetic fusion of the RBD to the nanoparticle trimer, and then we add in the pentamer to drive assembly. You get monodispersed nanoparticles like we're accustomed to at this point. And so I'm showing just a very truncated set of data here. This is from the first immunogenicity study we did in mice, where we saw that the nanoparticles elicited very high levels of neutralizing antibodies after 2 immunizations, whereas pre-fusion stabilized spike trimer elicited much lower neutralizing activity, and the monomeric RBD just didn't do anything at all. It's a very poorly immunogenic protein. And so this is a really great example of how you can take a small, poorly immunogenic protein and by multimerizing it, really transform its potency as an immunogen. The responses we were seeing were significantly more potent than a panel of human convalescent sera that we got from Helen Zhu here in Seattle. And we shipped our mice to Ralph Barrick's lab at the University of North Carolina, and Alex and his group challenged them with their mouse-adapted SARS-CoV-2 virus. And we saw that the nanoparticles at both high dose and low dose provided complete protection against viral replication in the nose and the lungs, whereas we saw breakthrough from the virus at low dose of the pre-fusion trimer. The monomeric RBD, again, provided no protection at all. And so, encouraged by these results, we continued. One of the potential criticisms of an RBD-based vaccine, of course, is that you're not providing the full complement of potentially protective epitopes that are present in the spike trimer. And early on in the pandemic, people were really concerned about this. And so Lexi set up a competition BLI assay that we could use to measure, are we making responses against multiple epitopes in the RBD, or are we getting just a very kind of non-diverse oligoclonal response? And what we saw was very potent responses against 3 distinct, non-overlapping epitopes known to be targeted by neutralizing antibodies. And so we felt comfortable that, it's not like we're hitting just a single epitope here. We are getting responses against multiple regions of the protein. And so hopefully that should minimize the possibility of viral escape. So this vaccine is in 2 clinical trials, a phase 3 at SK Bioscience and a phase 1/2 at our spin-out company, Icosavax. I'm showing here data from SK from their phase 1/2 trial that showed, in terms of antigen-specific antibody and neutralizing activity, significant increases over the NIBC standard panel of human convalescent serum. And so these are quite potent responses, and the vaccine also looks safe. So this one is in phase 3 and is advancing rapidly. Although I should note that Icosavax released their phase 1/2 data just this morning, or at least the top line results, and it did not look nearly as good. So something happened, and we need to figure out what, but I think this points to the complexity of biologics manufacturing. And I'm really glad that we were able to take 2 shots on goal here, one of which succeeded, and one of which seems to have run into a speed bump. So now that the SARS-CoV-2 vaccines are they are very close to being exactly the same. So the 2 groups are using 2 different adjuvants. So SK is using AS03 from GSK. Icosavax is using MF59 from CIIT. So that is one difference. They're also using slightly different processes. And in vaccine manufacturing, there's a catchphrase: the process is the product. And I think that may be what we're seeing here. Unknown, and investigations are, of course, starting now. But they're very, very, very similar vaccines. Yeah. So, of course, now that So in the phase 1/2 data, that was after 2 doses. SK has now gone back and given a 3rd dose to a subset of the phase 1/2 volunteers. And they're also doing that to some of the phase 3 subjects as well. And that's, of course, in response to Omicron. So many SARS-CoV-2 vaccines have seen dramatic reductions in neutralizing activity against Omicron, but that seems to be taken care of by a 3rd shot, right? So Moderna, Pfizer, all of these have seen the same thing. We have unpublished data in NHPs showing the same for this vaccine. So I'm sorry? Right. So the RBD that's in this vaccine is just the Wuhan 1 ancestral strain. And so 3 shots of that Wuhan 1 RBD nanoparticle brings up the Omicron neutralization almost to the level of neutralization against ancestral virus. Not quite. There's still a slight reduction, and that's also true for Pfizer and Moderna and everybody else. Yeah. But it really seems, of course, now in the headlines, we're debating a 4th shot, right? But 3 shots for these vaccines, all of them, is a good idea for protection against Omicron, for sure. And ours is no exception. Yep. So, of course, now we're starting to think about other coronaviruses, right? So SARS-CoV-2 is the 3rd beta coronavirus to make the jump to humans and cause significant problems in the last 20 years. There was SARS-CoV, there was MERS, now we have SARS-CoV-2. So this will happen again unless we do something to prevent it. So the idea is, can we design a vaccine that provides broadly protective immunity? In this case, what we're doing is we're going after sarbecoviruses, SARS-like coronaviruses first, right? And so we're taking the same mosaic nanoparticle approach that we did in flu, where we produce and then co-display multiple RBDs on the same nanoparticle. We have made 2 versions of these. We published a prototype version last year, and then this is kind of a 2nd gen that we're doing now. We're doing a tetravalent nanoparticle that includes an RBD from each of the known clades of sarbecoviruses, so clade 1A, clade 1B, clade 2, clade 3. But the clade 2 RBDs are a bit different than the others. Their entry receptor is not known. And so we also made a trivalent because it's a little bit unclear how relevant that clade 2 RBD is. And then immunized mice were shipped again to the Barrick lab, and they challenged these mice with a virus called SHCO14 This is a mismatched clade 1A virus, so it's most closely related to SARS-CoV. But in the RBD, there's, I think, 18 or 19% of the amino acids are different. So this is a heterologous and relatively stringent challenge. And what we saw was that both the trivalent and the tetravalent provided robust protection against this stringent challenge relative to control mice. And so we're encouraged by these data, but we're getting additional preclinical data on these candidates. And our collaborators at SK have received funding from CEPI to move whatever lead candidate we select here into phase 1 and 2 clinical trials. So that's kind of what we've been doing in the vaccine space over the last couple of years. We've got a lot of other efforts going on that I didn't cover today, but our group is primarily, we're not a flu lab, we're not a coronavirus lab, we're primarily a technology development lab. And so now that we're kind of through the crush of the pandemic, we've been getting back to some of our technology development work. And I wanted to share just one brief story with you on this. So, you know, the image on the left here is a collection of the sorts of things that we've been able to design so far. So, you know, homomeric particles that are relatively small, and then these 2-component nanoparticles constructed from 2 different proteins that, you know, can be small or can be relatively large, let's say 25, 30 nanometers in diameter. But these all share the characteristic of having perfect symmetry, right? So they have either 1 or 2 subunits in the asymmetric unit, but then you apply perfect symmetry to regenerate the entire structure, tetrahedral, octahedral, or icosahedral symmetry. But viruses, of course, have developed ways to get much larger by breaking symmetry. So virus capsids often use pseudosymmetry or quasi-symmetry to take a small number of building blocks and generate very large structures. This isn't even really that big. This is an 80 nanometer capsid, but you can get, virus capsids can and do get much, much larger than this. So we wanted to see if we could take a first step towards breaking symmetry and designing arbitrary nanoscale protein objects. And we decided to go after a pseudo-symmetry approach. So Quentin is an extremely talented graduate student in the lab who's been leading these efforts. And what he's doing is breaking the symmetry of a homotrimer such that we have genetically distinct subunits in the A, B, and C positions, but at the backbone level, we maintain symmetry, 3-fold symmetry. And so that's pseudo-symmetric, and this is a pseudo-symmetric trimeric building block. What that does for you is it increases the addressability of each subunit in the complex. So previously, if we made a modification to the A subunit here, that same modification would show up here and here because all 3 subunits are genetically identical. But now we can address the purple subunit specifically, or the blue subunit specifically, right? So Quentin did this in a homotrimer that we had previously built a nanoparticle out of. And I'll show on the next slide why he did that. It turned out in the end that he wasn't able to get to full ABC. He was able to get to AAB heterotrimers and ABB heterotrimers. And this was confirmed by native mass spec and Vicky Wysocki's group. So the key was by using a trimer from a nanoparticle that we had already designed, we call it cannibalize. We could reuse the interface between the blue subunits in this nanoparticle to come up with what we call a penton. So now instead of a pentamer of 5 subunits, we have a penton of 5 trimers, or 15 subunits, that still has 5-fold symmetry and has uniquely addressable subunits at each of the points of the penton. And what Quentin then did was he used this, so he lied to Rosetta, our software. He told Rosetta that this was just a pentamer, that's all it is, and used our standard 2-component nanoparticle docking and design approach to make this pseudo-symmetric nanoparticle that comprises 240 subunits and is 5.5 megadaltons in molecular weight. So he ordered 3 designs using this approach, and one of those assembled to the target structure. You can see it forms quite distinctive structures in cryo-electron micrographs, and the Wiesler lab was able to get a single particle reconstruction to 4.4 angstroms resolution that shows that what exists in the real world, again, matches what Quentin made up in the computer. So there are slight deviations, slight rotations of the subunits here relative to the design model. It's still quite accurate, but we think that the additional degrees of freedom in this system relative to the perfectly symmetric structures that we had designed previously allow a little bit of slop in the interface here. And we're looking into that further now. But while Quentin was characterizing these materials, he saw all these beautiful particles in the micrographs that looked just like what he designed. And then we saw this thing. And we were like, what is that, right? And so we started calling this the waffle fry for obvious reasons. And one of the most fun days I've ever had in science was Quentin and I sitting at a whiteboard trying to figure out what this was. And we worked out that what must be going on is, if you recall, he had AAB heterotrimers, but also ABB. And if there's contaminating ABB in his preparations, that could form not a penton, but a diron with twofold symmetry. And those can be incorporated on the icosahedral twofold symmetry axes to give this larger pseudosymmetric structure that kind of resembles a T=9 virus capsid. So this thing has 540 chains, 71 nanometers in diameter, substantially larger than anything we had previously designed. And the Wiesler lab got a single particle reconstruction at 6.6 angstroms resolution that shows that that is, in fact, exactly what's happening. And so Quentin realized when he saw this thing that this was the first in a series, an infinite series of nanoparticles that you could get by expanding hexagonal lattices on the icosahedral facets. And so in order to do that, you have to add in homo B, a homotrimer of the B chain. So no additional design was required here. We just made a new construct, expressed the B chain, purified it, and saw and tested whether we could assemble these larger pseudosymmetric nanoparticles. And it turns out that you can. So when Quentin did this, we started seeing this T=16-like nanoparticle that's about 100 nanometers in diameter with nearly 1,000 subunits in it. And a single particle reconstruction, again, confirms that that's exactly what you get. And so this is really fun. And one of the things that Quentin noticed is that the internal volume of this structure is significantly larger than in AAV. So about 2 orders of magnitude larger internal volume than in AAV. So that's fun to think about, but let's not forget that this thing is really very porous and doesn't have, anything related to packaging in it. We've got a long way to go, but it's exciting to think about what you might be able to do with a particle like this. And so because there's an infinite series of these things that extends from T=9 onward once you add the homo B subunit, Quentin wanted to see if we could shift the distribution of particles that form by shifting the subunit stoichiometry in the in vitro assembly reaction. So each of these has a different recipe, if you will, of the 4 different trimers that make up these nanoparticles. And so maybe if you add more and more homo B, right, and less AAB, you could drive the assembly of very, very large particles. And so Quentin set up a series of in vitro assembly reactions to test this. And using dynamic light scattering, he was able to show that you get close to matching the expected trend, but you do see this consistent increase in size as you shift that stoichiometry. Suggesting, and he has a few micrographs that I didn't put in here of particles that must be T=25 and T=36 like particles. So very, very large. And then because the surprises just never seem to stop in this project, the next surprise that we got was the formation of cylindrical lattices. So if you look at these larger structures, again, you get these hexagonal lattices on the triangular faces of these particles. So the homo B and the homo C, which we can't actually see here, it's down there, subunits form these hexagonal lattices. And so if you add just homo B and just homo C together and nothing else, you should get a hexagonal layer that extends infinitely in 2 dimensions. But what Quentin saw instead was tubes. cylindrical lattices. So these things roll up and close when they assemble to form this new type of structure that we had never seen before. And it turns out there's a precedence for this with HIV capsid, which also has a tendency to form cylindrical lattices, tubes, all kinds of other structures, kind of like this. So this is, so it's detergent. The question is this in high salt? We use a little bit of detergent to guide the assembly here. And it turns out, and I didn't put this in the slide, when you slowly anneal the thing by dialyzing away the detergent, you get the cylindrical lattice. If you quickly remove detergent, you actually do get two-dimensional layers. So the architecture that you get here depends on the assembly conditions, the solution conditions during assembly. So we got this by accident. This was a total surprise. But of course, one of the things we're doing now is trying to go back and develop docking and design methods to get these types of materials intentionally. And in fact, Audrey, a graduate student in the lab, started working on that about a year ago. And then when she saw these, she was like, Quentin, you scooped me. So not entirely, because you got to do it intentionally for it to count. Well, so we have not seen them close off yet, but actually, so Chelsea, a postdoc in the lab, has been playing with these things. And one of the things that's really fun, she's been able to assemble. So you'll see these are like 2.5 microns long, right? So for us, that's pretty big. She's able to assemble these things, and then we think they're terminated by subunit exhaustion, right? And then what she does is she adds in fluorescently labeled subunits, and you can see them add to the ends of the existing cylinders, and then she adds more in a different color, and you can see that color added to the end. Yeah, kind of. Well, but one of the really interesting things, so microtubules and actin and many other structures have helical symmetry. And if I'm going to be snobby, I would be a little bit snobby about helical symmetry. It's like the dumbest symmetry you can have. If you just take two things and you put them together and you introduce any bending at all, you get helical symmetry, right? So it's like the trivial solution. But cylindrical lattices are actually the geometric requirements are much more exacting. And one of the functional differences is that you have threefold axes pointing out from the cylinder here. So they're kind of like carbon nanotubes, but made out of protein oligomers. And so you can do things like put hemagglutinin on them, right? And so we're putting hemagglutinin and SARS-CoV-2 spike on these things. I have no idea what is going to happen when you inject that into a mouse, right? Like, is this going to be a good vaccine, a terrible vaccine? I don't know, but we're going to find out. So it's just fun to be able to build these new types of things out of proteins. Yeah, like, so the underlying particles are like, let's call it 25 nanometers. And when you put the antigens on, it's like 45 nanometers. Yes. 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 So the question is, what size are those? And those are about in the quote unquote right size range for draining through the lymphatic system to lymph nodes, right? So we think that those are probably in a pretty good size range. But like, how is this thing going to perform? I don't know, right? Is it going to be retained at the injection site? And then how is that going to affect its immunogenicity? We just don't know, but we'll find out. So anyway, so this is one example of the sorts of tech dev efforts that we've got going in the lab. And this is like the very first baby step you would take, right? This was the most minimal breaking of symmetry that you can imagine, but it already opens up so much new design space, right? So as we develop methods and get more sophisticated and break symmetry more thoroughly, the 25, 30-year aspirational goal is that someday we'll be able to design truly arbitrary nanoscale protein objects, right? Smiley faces, maps of the Western Hemisphere, whatever. So summary, just the key points, right? We have these methods for playing Legos with proteins. We've used them as multivalent antigen presentation platforms, and it really does seem to work pretty well there. and that multivalent display can significantly increase the magnitude of the humoral response, like the monomeric SARS-CoV-2 RBD, but that also these mosaic nanoparticles may be a route to broadly protective vaccines, and we're working very hard on this now. But then I think the key point is that we've only scratched the surface. I mean, this is just the very, very beginning, right? And that new computational methods and approaches are going to continue to open up new technology platforms. It's a really exciting time in the field right now. So I want to thank the people who did the work. So key contributors in pink from my group, from the Wiesler group. We have core labs at the IPD that are filled with amazing staff scientists, and a lot of the SARS-CoV-2 vaccine development was done in the core labs during lockdown in 2020. Key collaborators in the Baker lab at the IPD, at the VRC, and also in Ralph Barrick's lab at the University of North Carolina. We do a lot of work very closely with the Bill and Melinda Gates Foundation, and of course our other funders. And thank you for your attention. It's wonderful to be here in person with you today. Yeah, it's a great question. So the question was, have we ever used the mosaic nanoparticles to try to characterize individual monoclonal antibodies to see if they are broadly cross-reactive? So Annie is a graduate student in the group that's looking at this. One of the ideas we have is that it's the geometry of the epitope presentation that on top of the mosaic effect, we think the geometry of antigen presentation also leads to immunofocusing on certain epitopes, and that perhaps it's bivalent ligation of both arms of an antibody that does that by providing extra avidity. And so she's been doing this. She's been taking antibodies and making mosaic nanoparticles and then asking whether we can detect avid binding to the nanoparticle relative to monomeric Fabs. And you totally can, and it's epitope dependent. So certain epitopes are bivalently ligated and you get avid binding. Other epitopes are too far apart to bivalently ligate a single IgG, and you don't get avid binding. Actually, I don't think we've done that experiment with the RBD nanoparticles yet, although we have sent them to Albert Heck. He's going to try to do the same thing and characterize it by native mass spec. Annie's mostly been doing this with flu. Yeah. And flu and RSV, actually, RSVF. Yeah. Question is, how do you decide what is the optimal scaffold for the nanoparticles? We don't know, and that I think is one of the key questions that we need to answer. So everything I showed you today, to be perfectly honest, was particles that we just built, and then we put the antigens on afterwards. So like, yes, we got these broadly protective responses in flu, but that was the first shot we took, and you cannot tell me that that is the optimal nanoparticle. It's like statistically impossible, right? There has to be a better particle out there. What does it look like? So that's one of the things we're trying to do now, is design systematic series of particles where we can very precisely modulate things like antigen display geometry, content of the mosaic array, and then put it into a functioning immune system and ask, what does the ideal vaccine look like, right? But we're doing that now. We're starting to get encouraging data, but we just don't know yet. It's a great question. I think it's the question. Right, right. So the question is, does bivalent ligation of the two arms of an antibody elicit different signaling or a better response? So this is something that I posed as a question to Annie, and she thought I was crazy. And I love that you asked the question, because I wonder, right? Can a BCR feel it when both Fabs are ligated, and does it signal differently? Correct. Correct. Totally. And I think Annie and I have talked a lot about this. I think what we need are some really awesome B cell engineers to help us with that. Because actually we have kind of crazy ideas that I would love to talk to you about there. But yeah, I think it's a testable hypothesis, and I think it'd be super interesting. I mean, if you think about like integrins, the signaling, right, is very subtle. You can detect really subtle differences outside the cell and transmit that information inside the cell. So although it's maybe a little bit crazy, I don't think it's outside the realm of possibility. Yeah, yeah. Yes. Yeah, yeah. So two questions. One, are the scaffolds immunogenic? And then second, are the porous ones maybe more immunogenic? So the answer to the first question is yes, definitely. So the nanoparticles got right. They're foreign proteins that are forming a particle, so they're highly immunogenic. We always measure those responses and we publish the results. So we're very upfront about that. You also see T cell responses against peptides in the nanoparticle subunit. So you're getting like intrastructural help from the scaffold, we think. But the next question there is, does it matter that you're getting immune responses against the scaffold? We need more data. So I'm just going to say that we need more data, and I want to see data in humans on this. But we've done experiments in mice where we pre-immunize with just the naked nanoparticle with no antigen on it to get really high levels of nanoparticle specific antibodies. And then we boost those animals with the same nanoparticle displaying antigen, and pre-immunized animals show no differences in the antigen-specific response compared to animals that didn't receive the naked nanoparticle pre-immunization in terms of antigen-specific binding titers and neutralization. So it doesn't seem like there's a deleterious effect, but again, we need more data to be sure. And the second question, are the more porous nanoparticles more highly immunogenic? Again, I love the question, and it's one I hope that we can address through design, but we just don't know yet. Yeah, thank you.
Is there anyone on the WebEx that has questions? If you can hear us? Other chat? You can talk to WebEx, ask questions. You were told this.
Here. Right. Yep. So right. So the question is, the mosaic approach, everything today was in naive animals. Will it still work in the context of pre-existing immunity? Critical question. So we have done some experiments preclinically to address this, where this is in collaboration with Masaru at the VRC, we pre-immunized non-human primates with a series of seasonal influenza vaccines, right? 2006, 2007, 2008. And then we come back and we boost them with either another seasonal vaccine or the mosaic nanoparticle. And we do see that in that context, and this is an imperfect model, right? But in that context, we do see the mosaic nanoparticle robustly boost stem-directed antibodies, whereas the seasonal vaccine doesn't. So that's one of the reasons we're looking towards the clinical trials for the flu vaccine to really address this question in humans with complex influenza immune histories. Yeah. But so we need more data. Yeah.
On the mosaic and active.
Mm.
Looks like it was when we looked at it without the comparison chart.
Yes.
Can you comment on that? If the mosaic is the adjuvant needed to bypass that one with the mosaic?
Yeah.
Interacting with germline configuration.
Yeah, it's a good question. How much do we need the adjuvant, and particularly with the mosaic? So in mice, so we've done a lot of experiments with these particles by now, and what I would say is generally in mice, you can get by without adjuvant and you can still get decent responses, but it does not translate to higher mammals. So yeah, so in non-human primates, every time we've gone unadjuvanted, the responses are very, very poor. So even though these things are nanoparticles, even though they are more potent than soluble antigen, you still need the adjuvant. Yep. Hmm. Yeah, so the question is, how easy is it to incorporate adjuvants into the nanoparticles? So we're just about to submit a paper where we've shown that we can encapsulate a variety of molecules inside the nanoparticle. And one of the proof of principle applications that we did in that paper was we packaged resiquimod, right? And showed that that can increase the immune response. And if you more so than if you provide free resiquimod, and it's got the systemic toxicity and all that, right? But I I'm kind of dissatisfied with that approach. I don't like the idea of taking existing adjuvants and packaging them inside. What we are trying to do is just completely replace those existing types of adjuvants by designing de novo proteins that engage the immune system and tune responses one way or another. And so the Baker Lab has actually just yesterday, a paper came out showing that the methods for binder design have matured to the point that you can pretty much de novo design a tiny little protein that binds to any protein you want, as long as you have a structure. And these methods are very robust now. And so the idea is, can we design proteins that bind to pattern recognition receptors or Fc gamma receptors or whatever, right? And then incorporate these into the nanoparticles as protein-based adjuvants. And so we're starting to move in that direction. We've got several projects ongoing, but it's still early days. Mm. Mm-hmm. Mm-hmm. Right, right, right. Yeah, yeah, yeah. So great question. So David is not convinced by the difference between mosaics and cocktails, and is there any more information on that? So I want to know, right? I really want to know. Is the mosaic hypothesis working the way that we think it is? And we're not going to get the answer in the types of experiments we've been doing so far. We've got to do more mechanistic studies. So one of the things that we're getting off the ground right now is we're going to be working with Facundo Batista, who is making all of these transgenic, fully prearranged knock-in B cells, right? And then you adoptively transfer them into wild type mice, and you can in vivo very easily track activation and expansion of specific lineages, right? And so I want to use that to evaluate systematic series of mosaic and cocktail immunogens and really directly ask the question, does a mosaic nanoparticle actually preferentially expand that cross-reactive B cell, right? Right. No, I mean, I think ideally what we would want is we want that cross-reactive B cell to go nuts and affinity mature towards this highly conserved epitope and then differentiate into a plasma cell and spit out antibody for the rest of your life. But we got to figure out how to make that happen. Nope, we did not. Right. We have not looked at IgM. We need to. And then the other thing that we really need to do is just clone out a bunch of monoclonals from these studies, right? And just make 100 from the cocktail immunized NHPs, 100 from the mosaic immunized NHPs, and count the number that are cross-reactive. Important. Yep. Definitely. Definitely. Yep. So there's a lot. So the language that I use, right? There's a subtle yet consistent edge, which I think accurately characterizes the performance of the mosaic, but I am not yet convinced. I want direct evidence that either supports or refutes the mosaic hypothesis. I'm not satisfied yet. Yeah. Yeah. Right. Yeah. So the question is, have we looked at the memory compartment and the difference between cocktail and mosaic there? The answer is no, we haven't. We should. Um, we tried to get at that a little bit in the pre-immunity studies that we've done in NHPs, but the number of animals is so small that it's hard to really know if we're actually seeing differences there. So we don't have great data on that yet. Yeah. Right. So the question is, what are the goals of making the larger particles? So, yeah, I think there are lots of things you could imagine doing with them, right? So having higher valency display, being able to put more things on the particle is inherently useful. I didn't talk about it as much there, but because there are 4 different trimers in specific and distinct locations on the particle, you can start putting things in specific locations, right? Let's say you wanted to co-display 4 enzymes to reconstitute a biochemical network on the outside or frankly the inside of that particle. You would have 4 different locations where you could put enzyme A here, B here, C here, D here, right? So I see it as really, right, copy number, size, and addressability are kind of the proximal advantages of those particles. But really, in my mind, that whole project is just one baby step towards making fully asymmetric structures, right? So ATP synthase, right? The C ring has symmetry, your alpha beta subunits have symmetry, but the rest of the thing is asymmetric. And that's what makes it such a wonderful molecular machine, right? So right now we cannot design ATP synthase because you have to design 17 different protein-protein interfaces, and every interface has a failure rate associated with it. We'll try 10 things and only 2 of them work. And if you're doing 17 at the same time, zero point 0.2 times zero point 0.2 times zero point 0.2 times zero point 0.2, 17 times is a very, very small success rate, right? So we need to continually improve our computational methods to the point where we can progressively design more and more asymmetric structures. And this was just the very first step. Yeah. 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 So the question is, have we ever considered using these as drug delivery vehicles? So I never thought I was going to work on vaccines. Delivery was actually the thing that I've always wanted to do. That's what's been driving me for the last 20 years. So yes, we're definitely thinking about that. And for me, I'm more interested in delivering biologics rather than small molecules. We do have some small molecule projects going on in the lab right now. Um, but I think there are multiple challenges to delivering biologics. And honestly, packaging is not the big one anymore. So, we can package things in particles like this, but it's targeting specific cell. Well, just getting to the tissue and the cell that you need to get to in the body is in and of itself a major, major challenge. And then, targeting that, binding to that specific cell and not others, and then entering the cell and then crossing the membrane to access the cytoplasm, and then disassembling and releasing cargo. All of these are very, very difficult challenges. And that's leaving out the big one, right? The big one for using proteins as therapeutics is immunogenicity, right? So immune responses against your drug delivery vehicle are going to wipe you out, right? And that was part of the reason that we started working on vaccines in the meantime. Immune responses are great, right? That's exactly what you want. So you're swimming with the current instead of against it. And what we're hoping is that we can learn through the vaccine work how the immune system perceives these materials, and then hopefully be able to modulate that in the context of delivery. But I think that's a long-term goal. Mm-hmm. Great question. So the question is, in the two component particles that we showed so far, one of the proteins is made of mammalian cells, one is bacterial. Does it have to be that way, or do we have fully mammalian that you could deliver on a nucleic acid? So it is not out of necessity. We did that because making proteins in E. coli is cheap, easy, and robust. So for scalable manufacturing, we decided to stick with the E. coli for that pentamer. We do have particles that are fully mammalian. And in fact, one of the things we've been working on launching these from nucleic acids for years, actually, well before the pandemic. We just weren't ready in time. But we do have nanoparticles now that can be encoded on a nucleic acid and just pop out of cells pre-assembled. And so we are exploring those as mRNA launched nanoparticle vaccines, and we're doing a lot of technology development in that space right now. Yeah. We will have data. We will have data very soon. Yeah. Question is, can we put fusion machinery on these things to enter cells? So we have done that in the context of a different platform that I didn't talk about today. So in another project, we engineered these things to assemble inside mammalian cells and induce their own release inside of cell-derived membrane envelopes. And so they look kind of more like enveloped VLPs at that point. And there we have put fusion proteins in the membranes and then driven membrane fusion to enter cells. For the non-enveloped ones, we have not tried that yet using like non-enveloped viral entry proteins. The entry process is much more poorly characterized, and we haven't really gone there yet. But there are a lot of efforts at the Institute for Protein Design and in David Baker's lab in particular to design proteins for membrane traversal. Yeah, but it's a tough one. Thank you.